Before, just a minute. Okay. Before we proceed any further, I would like to remind everyone that our esteemed speakers are given 20 minutes to present their papers, and there will be another 20 minutes for the Q&A at the end of the session. Towards the end of their 20 minutes, I will be reminding, or our MC will be reminding each of the speakers when your time is almost over in just 2 minutes before your time is up. Please understand that we have a tight schedule, so we have to follow the timeline, the timetable. After all our resource speakers have presented, the participants will have their chance to shoot their questions on the papers or on the presenters. So in summary, we will have 1 hour for the 3 presentations at 20 minutes each, and another 20 minutes for the Q&A. We would also like to give our resource speakers the chance to give suggestions in relation to Biotropes program and activities and for future research endeavor in the Southeast Asian region. For our 3 topics and 3 speakers this morning, for topic 1, it is about education curriculum for biodiversity and environmental materials for high schools and vocational schools, to be presented by Mrs. Shifa Torsina. Topic 2 will be on the introduction, sampling, data collection, and uses of flora biodiversity, to be presented by Mr. Eddie Permana. And the third topic will be on the introduction, sampling, data collection, and uses of fauna diversity, to be presented by Ear Trihero Widarto. Our first speaker hails from Bandung, Indonesia. She is a teacher at Rongga Vocational High School, and she is a graduate of bachelor's degree in physical education and has a master's degree in master in management. Currently, she is a teacher 1 of Rongga Vocational High School, and currently the chairperson of the Kabupaten Bandung Barat Vocational High School Physics Teachers Conference, and a board member of the Indonesian Teachers Association, Board of West Java Region. Our second speaker hails from Ciamis, Indonesia. He has a bachelor's degree in forestry, major in forest management. He has over 30 years of experience conducting field survey either on wetland or in dry land, on mangrove, heath, lowland, or hardwood wetland. Okay now. His expertise is in plant biodiversity assessment, environmental compliance, including the establishment, inventory of permanent sample plots, plant identification and herbarium collections, forest rehab, revegetation, and restoration of post-mining areas. He used his expertise in the implementation of land rehabilitation, environmental assessment, and planning in the upland and lowland forest areas. His duties also include collaboration and communication with local communities, local and national government institutions, local, national, and international research institutions, consultants and universities, and higher education. Outside Indonesia, his biodiversity studies were in Chiang Mai, Highland, Bangkok area, and on the mangrove and coastal areas after the tsunami that hit in southern Thailand. He has a wide range of work experience as an ecologist, forest biologist at Private Hatfield, a Canadian environmental consultants group, as a lecturer of dendrology and forest ecology at the Faculty of Forestry, Bogor Agricultural University, as a field survey coordinator at the Center for International Forestry, as the senior advisor for environmental compliance and external relations at Private Weda Bay Nickel, as the senior biodiversity advisor for vegetation and restoration activities at Private Mears Suputan Mining. And at the start of 2021 up to now, he is a part or a member of an expert team at Private Primakilola EPB University. By the way, right after graduation, he was a research staff at Chemio Biotrope. Finally, our speaker is a prolific writer and has written and presented a lot of scientific papers in his field of expertise. So that's our speaker number 2. And our speaker number 3 hails from Purwokerto, Indonesia. He is currently a lecturer at the EPB University in Bogor. He finished his bachelor's degree at EPB University, Bogor, and his master's degree from James Cook University, Australia. His topic is on the introduction, sampling data, collection, and uses of fauna biodiversity. Our speaker is Mr. Ir Trihero Widarto. He is our third and last speaker, the last but not the least. So for now, we are going to start with our first topic to be presented by our first speaker, Mrs. Shifa Torsina from Ronga Vocational High School. Mrs. Shifa, the next 20 minutes is yours.
Thank you to the moderator who have given me a chance to deliver about education, curriculum for biodiversity and environment, materials for high school and vocational school. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Salam, Om Swastiastu, Namo Buddhaya, dan salam kebajikan. Without lingering longer, I will soon start to talk about the speech I am going to deliver this time. Can help me to show my...
Yeah, please share her screen. Please help her share the screen. IT people, can you please help Mrs. Shifa to share her screen?
Yes.
Yes, we are working on that. Our IT is still trying to perform the slides. Wait for a second. Thank you.
So our first speaker has experience in teaching vocational school students in the area of biodiversity, and so we are going to learn from her how the school children learn about biodiversity and how they enjoyed, how the teachers made the learning fun for the children, especially on topics like biodiversity. Okay, Mrs. Shifa, time is yours.
Yes. Thank you. Another slide, please. Yes. This is overview of One Rongga Vocational High School. At One Rongga Vocational High School, there are 3 majors, namely teknik dan bisnis sepeda motor, rekayasa perangkat lunak, dan agribisnis tanaman pangan dan hortikultura. Vocational school who focusing on human resource development in agriculture and technology. Next. Why is biodiversity so important to us? We believe that biodiversity is very important for the balance of Earth ecosystem. Besides that, we also believe that humans live well because of biodiversity. Nowadays, biodiversity is increasingly being threatened by its existence. That's why we care about biodiversity and environment. SMK Negeri 1 Rongga has an environment consisting of parks, fields, and an environment in the form of land that can be used as a source of learning, especially to get to know the concept of biodiversity. How application of biodiversity and environment learning at SMKN 1 Rongga? Biodiversity learning by utilizing the school environment can motivate student learning and achieve fun learning activities. Learning is more meaningful because students can deal directly with real situation. Types of cultivated plants that can be harvested or cannot be harvested related to the very diverse biodiversity of species that live within the scope of agricultural ecosystems can be an indicator of ecosystem health. For this reason, school can take advantage of the school environment to serve as a place for biodiversity. Yes, we plant in school, we plant orchids, aglonema, miana, calathea, lilies, mini cacti, but still in small quantity. We have also participated in the food. We also have vegetable hydroponic. What is the problem? There is 3 problems. The first, focus on food, biodiversity, and environment material itu fokus on food, crops, and horticulture agribusiness department. In yang second, 2013 curriculum, biology learning is only available in the food, crops, and horticulture agribusiness department, and there is not available in the motorcycle engineering and business and software engineering department. The last, Merdeka curriculum with the subject of social science project and project to strengthen the profile of Pancasila student. There are no modules or books that facilitate material on biodiversity and the environment. What is the solution? Next. For 2013 curriculum, we plan inter-subject teacher will design learning with the theme of biodiversity and the environment. For example, collaboration between physical education, sport and health, story, and Indonesian subject. And di sini kurikulum 2013 tidak ada untuk kelas 10 dan kelas 11 saja, karena kelas 10-nya sudah memakai kurikulum merdeka. Jadi untuk kelas 11, kelas 12 karena tidak ada pelajaran biologi lagi, tidak ada pelajaran project IPAS, maka kita mengambil collaboration teacher-nya itu antara mapel yang saling berhubungan. Next. Ini untuk yang kurikulum 2013, because ada mapel project social and natural science and project penguatan pelajar Pancasila. The teacher will make teaching modules related to biodiversity and environmental materials for project in the 10th grade even semester of all majors. Itu juga yang akan dilakukan untuk nanti proyek penguatan profil pelajar Pancasila. We have also participated in the food security program. Next, please. Next. Ini the role of school in food security. We and the community through the school community have collaborated in the development of butternut cultivation. Because ini adalah program kami dengan komite sekolah dan masyarakat untuk ketahanan pangan dalam penanaman butternut. Next, please. We hold strawberry cultivation too, and next we hold orange cultivation. Rongga area is an ecosystem support for the so in our area there is a new hydropower development, namely the Upper Cisokan hydroelectric power plant. Related to this, we who are in the hydropower environment want to help preserve the nature of our area. We will educate the community doing tree planting, joint cleaning the environment with the community. Next. Nah ini SMK Negeri 1 Rongga berada di dekat dengan bendungan Cisokan yang akan dibuat untuk PLTA. Next. How can students be aware and careful of the importance of biodiversity and environment? Not only theory and student activity in learning biodiversity and environment, but the most important thing is how we instill a positive character of caring the environment in students, and all of that cannot be done by one teacher, but there must be collaboration between all school residents and all stakeholders. I believe that students not fully don't care, it's just that they don't trust, don't believe in themselves, friends, teachers, family, and their environment. For this reason, as an educator, teachers should be able to build the trust of the student. One of them is by encouraging the students' voices so that students believe that their voices will be heard, meaningful, and can change the world. We can do this by training and doing student agency habituation to students. That's all that I have delivered, and I'm saying thanks a lot for giving me chance to involve in this equation. May Allah bless all of us. Wassalam.
Thank you very much, Mrs. Shaifa, for this very informative and enlightening as well as insightful presentation. Indeed, biodiversity in relation to food security is very important. And also equally important is to make this learning and understanding available for our school children. It's very important for the young minds to understand what is biodiversity all about and its importance. Thank you very much. So now we proceed to the next speaker on the topic on introduction, sampling, data collection, and uses of flora biodiversity. Thank you. Mr. Eddie, time is yours.
Terima kasih. Selamat pagi. Good morning, everybody. So, I'm Eddie Permana. So, I will take a topic for this presentation is introduction, sampling data collection, and uses of flora biodiversity. Before of that, I'm, everybody, you see my clothes. Yes, my address is using batiks. Batik is Indonesia traditional, clothes. So in the batik show, Papa is also using a batik, wearing a batik. So batik is, you see that is a miniature of biodiversity. So like my shirt, this is Lombok batik. Batik is, there are some, this is a flower of Sesbania grandiflora or turi. And then also there is another, this is also the leaves of Sesbania grandiflora. Grandiflora, turi is like food, so that is for herbaceous. Okay, so my presentation is, will be starting right now. Okay, Indra. So this is, I think there are 35 of slides. So maybe I will skip if because theoretical. So maybe Bapak, Ibu, everybody this will be more interactive with using the how to analyze the data, right? Okay, so objective of the training is creating and improving trainees' awareness on biodiversity, then to improve or calculate the quantitative data analysis of flora diversity, contribute the effort and endeavors of sustainability for conservation. Next. So, biological diversity is that, I think this is a definition, right? Yes. Yeah, biological diversity. So maybe everybody can see that it's the definition of biodiversity is like diverse of ecosystem species and diversity and also the genetic diversity. So if you see on my left, so that is there's also many plants outside of this building, so there are many plants, and so that is also like a miniature of biodiversity. Okay, next. So you know, Indonesia is the world's second largest biodiversity or mega biodiversity. So why is called mega biodiversity? Because rich of biodiversity and then have many flora, fauna, endemic species. So we can see the compare with other countries in the world. So next. So we say Indonesia is number 2 largest biodiversity. Number 1 is Brazil. Brazil is a continent, American continent. Indonesia is island. So there is it's not so like a continent, yeah. Even that the diversity is more like that. So because Brazil and Indonesia is same tropical countries, so I think we cannot compare with another with the 4 seasons of diversity. Next. So you can see also the wide group of flora and fauna in Indonesia. So country with biodiversity is a total of 27,500 species of flowering plants, then 515 species of mammals, and then 1,539 species of birds, and then reptiles and also amphibians. So you can see that in the table. So Indonesia can compare with another world. So I think it's about 10 or more than 10% of Indonesian is why it's called mega biodiversity. Next. So function of biodiversity is a source of livelihood and survival of human beings as material for clothing, food, shelter, medicine, and other necessities of life come from there. Then source of science and technology, development of social and cultural humanity, and the decline in biodiversity is causing the loss of human benefits that can be obtained. The decline of biodiversity can be prevented by doing conservation biodiversity. Next. So here is the methodology of data gathering. Indonesia is usually using this vegetation analysis. So vegetation analysis is a method to study about the structure and floristic composition of vegetation or plant communities. So vegetation, what is vegetation? Vegetation is a plant community which live on an area in the ecosystem. And then plant community is a collection of or association of plant species within a designated geographical unit which form a relatively uniform patch, distinguishable from neighboring patches of different vegetation types. And plant community is a consociation. So there are consociation and association. Like consociation is one type of community, like a pine forest, mangrove forest, and then others. And then association is a various of type of community, like grassland or alang-alang is a mix with a shrub. You can see next in the photograph later on. Next. Vegetation analysis is study of natural forest stand, regeneration stage from seedling, sapling, poles, and trees. So actually, why we use this call is like a regeneration stage, because forest is not like a plantation, right? This is a natural forest, I mean. So you can see in the forest, we don't know the age of the plant. So we use the stages, 4 stages, like seedling, and then sapling, and then poles and trees. So we can see next. So study of undergrowth plants, alang-alang or grass or savanna and shrub, and then study of vegetation, study floristic and then vegetation. So floristic study is like a qualitative data, like form, distribution, etc. So there is no number or something like that. So like vegetation is a qualitative and quantitative data analysis. So we can show if, for example, in 1 hectare, there are like 1,500 something like that of species of pine, for example, like that. And then quantitative data is a numbers of size, dry weight. Dry weight this is a biomass. And then area ground, measurement, and observation. Next. Now here's a plot shape. Plot shape is supporting the ease of vegetation analysis and sampling efficiency. Why? Because if we use the plots, so it's not necessary to inventory all of the ecosystem. And then various type of sample plots like a rectangular, square, and then circle. So for rectangular or transect, it's effective for study the changes in vegetation due to the influence of environmental change with the direction of perpendicular to the contour of lines. And then it's a comprehensive way to use because it can be adapted to all types of plant communities. That is for the square or quadratic. Then circle is a low vegetation, like is easy in practice by turning the group at the point of the circle. Measurement error is smaller, yeah, if using the circle plot shape. And then number of plots. So the minimum of number of plots to be taking into account the constraint of time, cost, and energy, but it should be fairly representative. Optimal size and number of plots representing how the plant community, like a species scope area. So if you talking about this species scope area, so it means if in 1 hectare, for example, we made some repetition of 10 to 10 meter square, like 100 meters square. So, 1 hectare is about 10,000 meters square. So if we repeat it every 100 meters, if the data still increase, so we have to continue until the data is stable. So that is it called a species scope area. Next. Description of vegetation analysis method. So number A, square method is a single plot, only one sample plot, area of plot based on species scope area, suitable for the homogeneous forest. Area plot, it's already tested by Mayer in 1954. The area is 2,500 square meter in the Terracotta forest in Bangka, and then Nicholson 0.6 until 1.5 hectare in North Kalimantan, and then Richard 1952 is 1.5 hectare for tropical forest. Tropical forest is a big, huge area. Yeah, right? Then Wyatt Smith in 1959 is about 0.6 hectare, and then Vestal 1949, approximately about 3 hectare. It's a very big, about the sample plot. And then Cain and Castro 1959, rectangular, 20 by 20 meter to 1,500 meters. So for the motivated by the square continuous plots. Next. Now here is the figure of single plot. So this is 40 by 40. So like I mentioned in Canextro. Next. This is a multiple plot. So there is a number of plots. That is more than one plot, evenly spread, right? Distribution is evenly. So like random, using random or systematic. Number of sample plots varied based on species scope area, vegetation condition, and sampling intensity. Regarding the oosting in 1942, for sample plot for tree is 10 meter by multiplied by 10 meters, and then undergrowth or shrub with the height less than 3 meters is 4 by 4 meters. And then undergrowth and ini yang lama. Okay, nah this is it. Yeah, for wild smith, for undergrowth and herbaceous is 1 meter by 1 meter. Then wild smith in 1959, tree is 0.1 hectare or 1,000 meters per square. Then undergrowth and sapling 0.01 hectare. And then undergrowth and seedling is 0.001 hectare. Next. Now here is multiple plots. Like random like this on the left side. The left side is random, so it's not systematically. So you can see here is like for here and then here, and it is random. So to put, for example, this is a square of 1 hectare, so it's not systematic like this. On the right side is systematically in one line like this. But random is random. So so you can put the plots anywhere. So this is random. Next. This is line meter or transect. So it's usually is using the huge forest, less known the site condition, and then it's suitable to determine the vegetation changes based on changing environmental factors. And Cain and Castro, 1959, so width of the area is 20 meter and then length 1,000 meter. Maybe next will be see the figure. So intersect distance is 200 meter and then intensity sampling is 10%. 10% is, I think it's very high, yeah, for the big area. So in I think 1967, yes. So in Ministry of Forestry, that if making a inventory for the huge forest is about there are 5 transect minimal, and then intersect distance is 1 to 5 kilometers. You can imagine in one transect to another transect is 1 to 5 kilometers. This is very huge. Then transects, the sample plots are combination of quadrat and line of square, where a nested sampling is found within a larger sample plot. So the rest is called as a nested sampling. Next. Like this transect method. So in one subplot, there are 20 by 20 meters, but inside the 20 by 20 meters, there is smaller, like 2 meter by 2 meter, and then 5 by 5 meter, and then 10 meter by 10 meters. That is a regeneration stage of the flora. Like 2 by 2 meters is for the seedling, and then 5 by 5 for a sapling, and then 10 by 10 for poles, and then 20 by 20 is for trees. This is a subplot. So you can see that this is a subplot. So this is another subplot, and you can continue maybe for 1 kilometer. There are 1 kilometer is about 500 subplots. Next. Line square method, like modification of multiple plots and transect. And then modification multi-plots is jump 1 or more plots in transect. This is any kind of making a plots, yeah. So it's not continue, but you know, this is another is a jump 1. So 20 by subplot, 1 subplot, then jump 1, and then continue with making a subplot, and then continue again. And then a plot shape of rectangle, a square, and circle. So the size of plots is 10 by 10, 20 by 20, 20 by 5, and then vertical is its radian is 17.8 meter or 0.1 hectare. Next. Now here is a saline square method like this, also same like this. So 25 and 2 by 2, 5 by 5, 10 by 10, then 20 by 20. Next. Now here's a circle method. So you radial of the, so if you stand here, so you can pull the roof about 17.8 meter, and then make a circle like this, and then so you can take a data inside of the plots. So inside the plots, so you can collect the data like diameter trees and then height and or species name. So it is usually using in the plantation forest. So with using this circle method, you can get the tree potential like a number of individual tree or plants and then diameter and height, etc. Next. Here is a variable area method. I was used this method while I'm working in C4 in 1920. In 2000, in 2000. And this variable area method is we're using there are two method of spot. Like this is for the small plant, with the 5 meter wide, and then length is 40 meters. And then while for the tree plants, maximum of the on the cell is 8 for trees. But this is maximum of the area is 40 by 40. So this is like one. This is maximum. Here is 20 meter, and then also maximum also like this is 20 meter. So there are about 4. Yeah. So this is it's not usual using in Indonesia, but we are using this method because using the rapid biodiversity assessment. So like in small landscape, so we rather than using the big area of plot, so we're using this variable area method. Okay, next. Here is maybe Bapak Ibu sekalian, this is usually business as usual in Indonesia with using this method. So if you want to make plot for the quantitative of data of biodiversity, we usually using like this nested sampling, with for A is 20 by 20, 2 by 2, and then B 5 by 5, and then C by 10 by 10, and then here is 20 by 20. So you can continue this subplot, for example, 200 or 500 meters. So with using this, so you can get the data with the floristic diversity, richness, and natural and anthropogenic distribution regime. So next. Sequence of flora data collection step. So if we already using this figure previously, so we can step field of data collection, like to determine the coordinate of the location of observation based on satellite imagery, and then record the location and surrounding condition information using the set description sheet form. And then sample plot are combining of quadrats and line of squares, where within the larger squares there are smaller squares. It's nested sampling. So for 3 sample plot for 3 poles, sapling and seedling stages, and then record data into the sheet form. Next, step of field data collection is in subplot A, record the seedlings of trees with vegetation with a minimum of tulips, and then height less than 1.5 meters, and then parameter recorded, number of individuals and plant species name. Then in subplot B, record the sapling with a height more than 1.5 meters, and then a stem diameter less than 10 centimeter, and then parameters recorded is number of individual and plant species names. In subplot C, record and measure pole regeneration stage with a stem diameter between 10 to 20 centimeter, and then parameter recorded is diameter, height, and name of plant species. In subplot D, record and measure the tree regeneration stage with the diameter more than 20 centimeters. That the parameter recorded stem diameter, height, and name of plant species. Okay, next. Now here is what is to be recorded like this. So beside the regeneration stages for trees, like tree seedling, yeah, in the 2 meter by 2 by 2 meter, and then 5 plants, and also we record also non-tree regeneration, yeah, like in the 2 by 2 meters, we record vegetation like shrub, herbaceous, fern, etc. And then for 5 by 5, liana, non-woody, parandanus, epiphyt, and palms, more than one, palms with long or length or height is more than 1.5 meters. But in the 10 by 10 meters, we will not record anything except the pole trees. And then 20 by 20 is, you can see the strangler and woody liana also we recorded. Maybe in the next picture, maybe we can see how to record it. Okay, next. Now here's when we already record everything about the plant. So and for data analysis, there is it's an important value index. Maybe next Indra. Here is before to the important value index. We can see the formula like the density. So density is number of individuals area of a sample plot divided by area of sample plot. So like for seedling stage, sapling and pole and trees, it will be different. Yeah. For example, for the area of sample and also number of individual and sample, because this is for example, for the seedling is 2 by 2 meters, and then sapling 5 by 5, poles 10 by 10, and then tree is 20 by 20. So besides the density, also relative density. So relative density is, so maybe this is a formula, yeah, formula, while you already record all the data from the forest, and then you can using this formula. So like density and then frequency. Frequency is number of subplot found of n species and divided by total number of subplot. So for example, in 100 per meter, there is 5. So 5 subplot. So the number of subplots found in, for example, in because there is 5. So for example, in one subplot, there is for example what you call is one species, yeah, like Mangifera indica, for example, Mangifera indica or mango. So if you find even in subplot there are 2 mangoes, so we only record one individual. So if in 5 subplots there are 5, so we'll be using recorded with this formula. So 5 divided by 5. mean is one. One mean, so in every subplot, there is always find that species. Okay?
Excuse me,
So,
Mr. Eddie. Could you please wrap up in
okay.
the next 2 minutes?
Okay. Okay, next. Now here is that is for formula and data analysis, and also this is diversity index. So we can see the maybe you can find that in the slides. Next, and then here's a data sheet form. Data sheet form that using while you are recording in the field. Next. So okay, if you see there are 4 pictures. Yeah, there are 4 pictures like this. This is. Anybody knows this is what this is area ecosystem? This is mangrove, yeah. Mangrove. Why? Because here there is aerial fruit, aerial roots. Yeah, I don't know. Maybe in Philippines it's called bakau lalaki ya, atau Rhizoptera apiculata or micronata. And then here's the beech forest. And then here is a peat forest. You know, peat forest. The water is very dark. Yeah, it's more than like tea water, more than. So it's like a blackie, yeah, like this. And here's lowland area, lowland tropical forest. So we can see this is asplenium. There's also this is here's. What's called is woody liana, yeah. So mean that is in the forest area, in the tropical forest area, the soil is not fertile, yeah. So this is why, because there is this is usually you can find also like orchid, something like that. So next. Here is a plantation, and then also this is plantation. This is alang-alang and mixed with shrub, and then this is also is I don't know the species. I forgot the species. The next. Now here's a product of biodiversity or non-wood. Like here's you know in Indonesia it's called rempah-rempah. Rempah-rempah is spices. And then also this is paper, and then also this is fungi. Next, here is a resource of biodiversity, like resource like this. Here's there is a orchid, birds, and then also houses. Yeah. Okay, next. Here's birds and fashion. Yeah, like here's a paradise bird. Yeah, paradise bird is very expensive and also endangered species. So this is a story about the paradise apoda. So it's called in botanical name is a paradise apoda. So if we translate, so paradise is a paradise is like a paradise. Apod means no food. So this is I think, I don't know, there is something wrong, yeah, when identification the name. Okay, next. Here is, you know, Yoda, you know, everybody maybe knows there is a movie Star Wars, like maybe this Yoda is maybe inspired by the tarsius animal from North Sulawesi. So, you know, this tarsius is a tiny primate. So maybe it's smaller than, it's maybe like, for less than 10 centimeter, yeah, the size. And then the conservation status is protected and vulnerable and endemic species. Next. Okay, that is all of my presentation. Thank you very much.
Thank you very much for another informative and enlightening presentation, Mr. Eddie. There's really a lot to learn from your 30 years of experience, yeah. So anyway, there will be questions and to be answered later on.
Okay.
So shall we proceed since we are running out of time? Our third topic is on the introduction, sampling data collection, and the uses of fauna diversity, to be presented by Ir Trihero Widarto. Mr. Trihero, the next 20 minutes is yours.
Okay, thank you very much. Can you hear my voice?
Yes, loud and clear on my side.
Okay, thank you. Good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Okay, I'm glad to be here with you to introduce about biodiversity and how to do sampling and data collection, especially for animal. I think we already know about biodiversity because the two speakers before has mentioned about that, and maybe in 2 days you already understand about biodiversity. That's very important for our life. So when our biodiversity it's experience destruction or broken, then maybe we human being will not be exist anymore. So that's very important for us to conserve the biodiversity as much as possible. Either the genetic diversity or species diversity or ecosystem diversity. I just want to show you this picture. This is Borneo from 1950 to 2005. Here, it's already changed a lot. The green area very diminished, and it will be predicted in 2000, and this is in 2020, the prediction. I haven't got any information about the real or the fake condition in Borneo, but this is the prediction in 2020 by NRDC. You can imagine if the destruction of our biodiversity, not only in Indonesia, but in our area in Southeast Asia, then we will have more problem in the future, especially for you, the young generation. For me, maybe in 5 or 10 years, I will go. As you can see here, only Indonesian forests contain a lot of mammal, birds, plants, comparing to the world. So then, actually, previous speaker, Mr. Eddie, already mentioned about the species richness in Indonesia. And this is the effect of overexploitation by trade. This is mentioned by Mrs. Clarissa in the previous meeting. There are 30 species of birds that endanger, 4 for flowering plants, 60 species of mammals, and so on. About 105 organisms. It's in endangered situation. So, we already know that we need conserve the biodiversity as much as possible, as soon as possible, before being late. And this workshop, the purpose of this workshop is to introduce the importance of biodiversity to the young generation as early as possible to the high school and vocational school students. So this is a good effort for conserve not only the fauna, but the whole biodiversity. So what is the first step we need to do to conserve the biodiversity? I think you know the answer. Yeah. That is, we have to know what is the size of the population now, or the existing population. How we can know the existing population by studying the population ecology. So we have to know the initial size or the existing size of the population. Then we have to do sampling, because we cannot do the to do census or to count the whole number of the population. Can you imagine if you go to Ujung Kulon and then maybe Ujung Kulon is still small area. If we do survey or counting the rhinoceros in Ujung Kulon by helicopter or through small plane, but if the area is very big, then we have to do sampling. The reason we're doing sampling, because we have a limited time, we have limited energy, cost, and resources. So we do sampling, but the data we collected from the sampling can representative the condition of the population, the number of the population. We can estimate, we can predict how many of them still there in the habitat or in the ecosystem. So we have to design the sampling so we can get the data effectively. When we design sampling, we have to be sure that the study sets are representative of the range of local environments. Secondly, the sampling of environment is replicative. This is a must. We have to do replication. And we have to have enough field time. We have to have enough time to apply sufficient and comparable survey effort at each site. If we have many sites, then we have to spend or spare enough time. So the data we collected should be sufficient to be analyzed. And also we have to spare time. Sorry, we have to choose optimal period to do sampling. It means that if we do sampling in the wet seasons, will be hindered by the climate, then we have to choose other time. In the dry season, for example. So we can get the data very optimum. And then we have also to be sure that our survey or our sampling, our work or our research can be repeated by other researchers or practitioners later. So in sampling design, first we have to have a study set that representative local environment. And then we have to make a layout of the sampling point. We already seen from Mr. Eddie before about layout of the sampling point. There are random sampling or systematic sampling or stratified sampling and so on. Don't forget replication, and then doing survey and timing and duration of survey or sampling. We have to calculate when we have to do the sampling, or during the day or during the night, depend on the animal we want to sampling. Of course, nocturnal animal, we have to do sampling at night. And then it's already mentioned that the survey or the data we collected are repeatable. Okay, there are some sampling methods. For vertebrate, we have two types of techniques: capture techniques and non-capture technique. We have to make a standard procedure when operating the traps that we are going to use to capture the animal. So we make the procedure standardized first. So when we use the procedure in other area in other time, then we can compare the result of the collection. We can do by hand, the capturing the animal, because, for example, if the animal hiding under the stones or under the big clock, then we have to use our hand. We can use dry pitfall traps. We can use funnel traps, box, and so on. And we have also to be careful when handling and releasing vertebrates. For example, usually the animal or large animal, macrofauna, when we catch it, then we handle it, they will defecate or urinate. So we have to be careful. We have to be safe using glove, or don't forget to wash your hand after handling the animal. This is non-capture technique. Non-capture techniques, we can use the signs of the animal, tracks, the skates or the faces, the nest, burrows. We can use cameras, motion sensitive camera. We can use ultrasonic detectors, aural survey for frogs, audio recording, air tube, and so on. Include also, we can use the regurgitated pellets. This is the nuts. After chewing by kakatu. So we can use this to estimate, or to, after we got the data, then we can estimate how many kakatus in the area. And different kakatus, as we can see, will bite or chew different way, in different way. For invertebrate, usually we use pitfall traps, leaf litter samples. We collect the litter or the leaves spread on the ground, and then we search for the animal hiding in the litter. Light traps or black light traps, sampling the vegetation for invertebrate and so on.
please
Yes.
wrap up in 2 minutes.
Okay, baik.
Thank you.
So don't forget the abiotic factors to be included into your recording. Yeah. Salinity, pH, temperature, dissolved oxygen, wave action, turbidity, and so on. This is already mentioned by Mr. Eddie about random sampling, systemic sampling, and stratified sampling. Yeah. So at the end, we have to estimate from the data how many animals in one area. Yeah. We can estimate by counting. We can estimate population density, percent coverage, and percent frequency. And also we can now we can see the richness measure of the number of different kinds of organism present in an area. Evenness, that is comparing the similarity of the population size of each of the species present. What I talk just now, you can check in these references. This is fauna sampling manual for wildlife. And then what else? This is about on method of biodiversity data collection and monitoring of species and habitats. Yeah. Monitoring also need data by sampling. This is the interesting way because we can do it for high school or vocational school students. The important thing in introducing the importance of biodiversity to the students is we can do as simple as simple research, like the first speaker said that in her school the teacher only introduced the biodiversity in the one department, or not in the two other departments. Yeah. So we can introduce to whole student, yeah. For example, by planting the trees in the school area. So we can make a group student, yeah. Planting the trees and then look after the trees and so on. So even though they don't get biology, they can learn or they know about biodiversity as early as possible. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Mr. Trihero, for this noteworthy presentation, and reminded that we have limited time, energy, and fund and resources to do the survey, so it is necessary that we do the appropriate techniques, and thank you for sharing those techniques with us. So that's our last topic for this morning session, and so we are now going to proceed with the question and answers. So that's for another 20 minutes. But before we go into that, I would like to request the speakers, the resource persons, to switch on their cameras. And another reminder, we would like to give a chance or priority to those outside Indonesia to ask their questions and share their experiences if they feel like doing it, so that we also can learn from them. And their knowledge that will be shared can also help Biotrope in its mandate, in fulfilling its mandate. So we have noticed that we have participants from outside Indonesia, like the Philippines, India, Myanmar. So we give these participants the priority to ask questions and of course to give comments. So thank you very much, resource speakers. So now we proceed with a 20-minute question and answers. Okay. Participants, you can use your raise hand menu if you want to raise a question, or you may write it on the chat box if you are shy to raise your questions. But we do encourage you to speak or voice right away your questions. So we have here very able speakers, very rich in experience, very experts in their field. So we encourage you to ask questions and learn more from them, in as much as they share with us their most precious time. Okay, who would like to give the first question? Right, there is a question here from Mr. Abraham Lotivio from the Philippines. Yeah. So can we integrate biodiversity education in the primary education? Who would like to answer that? So we were talking about high school and vocational schools. Now the question is referring to biodiversity education among the primary students. So primary or elementary, right? We call them elementary also. So who would like, from our experts, who would like to answer that?
Can I answer the question, please?
Yeah, sure, Mr. Trajero.
Okay.
Go ahead.
Thank you, Mr. Abraham Lotivio. In where are you from?
From Philippines.
Philippines. Okay,
yeah?
Philippines.
Mr.
Yeah.
Abraham?
Yeah.
Please
It
unmute.
is.
Sorry, please unmute, Mr. Abraham. Okay, there you
Yes,
go.
I'm from the Philippines.
Okay, thank you. Okay. Yeah. I think we have to do so to introduce about biodiversity to the student of elementary school. But of course, we don't need to say or to explain what is biodiversity is, what is the importance of biodiversity. We don't need to do so. But the simple thing we can do for them or to teach them about biodiversity and the importance for our life by, for example, ask them to raise an animal, cats or dog or others. Yeah. Then also can raise or plant a flower in a bin or a pot, yeah, and so on. And if in the school they have an area for planting, they can plant vegetables and so on. So it's a must. So we maybe in the next session, Biotrope can or semi-Biotrope can make a program for teacher from elementary schools or junior high school. Thank you.
Thank you, very much, Mr. Trajero. Yeah, Biotrope officials, I guess, are listening. So, yeah, that's a good input. Thank you very much. Wow, how is that? Okay, so another question. So while waiting for others to pose their questions, I have one or two questions here. My first question is addressed to Mrs. Shaifa. Mrs. Shaifa, are there specific examples of learning activities that you observed that high school students in your school really enjoy doing and that really help them understand the true value of biological diversity? So just some examples that really the students enjoyed doing, and thereby they were also able to understand the importance of biodiversity. I know you have done a lot of activities for them, you have conducted a lot of activities, but those noteworthy activities wherein everybody enjoyed, the children most especially, and also the teachers as well.
Yes. Jadi saya dan teman-teman ketika mengajarkan, mohon maaf saya memakai bahasa Indonesia, mengajarkan biodiversity and environment, teaching the materials, the children dibawa ke kehidupan nyata. Jadi diberikan masalah dulu anak-anak, masalah yang ada di lingkungannya. Seperti misalkan ada beberapa tumbuhan yang sudah tidak ada sekarang atau langka, susah untuk dicari. Kemudian setelah itu mereka diberikan project bagaimana untuk membuat sebuah untuk memberikan solusi kepada masalah tersebut. Selain itu juga dalam hal lingkungan, karena di daerah kami itu pegunungan, banyak longsor, biasanya kami memberikan masalah dulu kepada anak bagaimana supaya longsor tersebut tidak dapat terjadi. Terus anak nanti disuruh untuk mengeksplorasi, mencari sumber kepada BPBD di kecamatan, mereka berwawancara. Setelah itu setelah itu mereka membawa hasil dari wawancaranya ke sekolah untuk kemudian diolah datanya sehingga nanti mereka memberikan solusi kepada rekan-rekannya bagaimana untuk tidak terjadi longsor dan bagaimana supaya tidak menjadi korban bencana longsor. Kemudian itu sih sebenarnya biasanya yang pernah saya lakukan. Terima kasih. Oh iya, dan mereka juga cukup senang sih sebetulnya, karena sebelum belajar saya memberikan dulu penguatan tentang karakter apa yang harus kepedulian mereka terhadap lingkungan. Jadi memberikan dulu semacam keyakinan kelas supaya mereka mau bertanggung jawab atas apa yang mereka utarakan dan mereka menjadi lebih peduli kepada lingkungan. Itu saja. Terima kasih.
Thank you very much. Oh, sorry. So sorry, but the English channel was not that clear. But anyway, I hope our Indonesian participants were able to get the explanation from Mr. Shaifa. So the next question, since I could not see, I haven't seen a raised hand nor a question on the chat box, may I proceed with my second question? So the second question is actually directed to our two gentlemen speakers, because they have similar, not similar field, but similar topic on the assessment of biodiversity. Sirs, based on your experiences, based on real life situations, are there, for researchers in the field or researchers to be who are listening to us here, are there important things to do or not to do, or things that must do and must not do while in the field conducting the flora and fauna inventory. So based on your experience, because sometimes the theory does not tell us about what is really happening or going to happen in the field. So for our seasoned speakers here, may we learn from them how they, especially the problems that they encountered in assessing biodiversity. So how did they counteract, or how did they face or survive this kind of real life situations in the field that are, say, causing delays or causing some concerns in assessment. Thank you very much. Yes, sir, please.
Maybe me first, Ms. Evelyn, part 3.
Please.
Okay. Formally, as advised, some like mining concession, and then also like private company. So actually, the problem is when we make a survey, it's about area. Area is large, and also some of them also in the forestry area, and then also in the non-forested area. I mean, non-area under the government ownership. So, like in the private or the private area, like for example, we found some species of plant that is in danger and critically in danger. So the problem is, how can we conserve? So that is the problem. So when it's depend on who is the owner of the land. So when the owner said that this is mine, this is not yours. So there is so that's my experience. Last month in Maluku, so I found the Lichia ceramica. So Lichia ceramica is included in the critical endangered in the red list of IUCN. So we are also, that is maybe the good thing is to get the plantlet, and then also planting in other area, in the conservation area, or also in the government area. So we can advise that its area maybe it's good for us as an example, and then if we can make like it's a miniature of arboretum, something like that. So you can put that species of plant that is included in the red list in IUCN. So maybe some species can be planted in that area. So beside also in I think there is in the private company in Indonesia, there are some they are willing to conserve the biodiversity, like so not only the planting the trees, like in the mining area, not only the planting, but also that there is that is like beside the site specific species area, that is also should have there is conservation conservation or something like that. So that is, I think it's good for the private company. That's my experience.
Okay,
thank you.
So you have to report it to the proper authorities. Is that
Oh,
right?
yeah, sure. Yeah, sure.
So whether the company would like to own it or not, the proper authorities have to be informed. Is that right?
Yeah, right. So even that there are some companies that is there is a money lender from outside, like from Europe, that using the biodiversity, like in under the IFC or World Bank. So that is that there is a regulation that is very hard. Yeah, it's very hard. So like I was working in the mining company that is with. The money lender is from IFC, International Finance Corporation. So, for example, I'm working in the performance standard number 6. It's not conservation, not loss biodiversity. So, how we can say that it's a loss or not loss if you don't have the data? So while I'm working in that company, so research association, yeah, like from LIPI or something like that. So I try to collect more data, so even in that area, I found 50 new species, right? And also there are 5 of genus.
Amazing, yeah.
Not only the plant, also there is also like rat also, that is also new genus. So with using that is in Halmahera, so there are so many species that is not open area. So that is my experience. Thank you.
Thank you very much, Pa Eddie. So we give floor to Pa Trahiro.
Okay, thank you. Yeah, I have experience when doing sampling in the water. I was collecting freshwater mussel in the river. I cannot dive, and the water is quite deep. So I have to find a way how to collect the mussel that live in the bottom of the river. So then I have to use my feet to collect one by one the mussel. So this is just example that we have. When we go to the field, then we have to make a good preparation, because if we go to the remote area, then if we don't bring proper equipment or chemicals, then we will have difficulties in doing sampling. But when it's happened, then we have to be creative and innovative in solving the problem. We have, if we have no rotan, then root is okay.
Yeah, thank you very
That's
much.
it. Thank you.
Thank you, Pa Trahiro. Yes, we have to be creative and innovative even in doing this field sampling. And in the Philippines, we call it discarte, yeah, Mr. Abraham? Discarte in the field. Especially so if you forget something very important
Yes.
as an equipment, yeah. So, any more questions from among the participants?
Okay.
This is your last chance to inquire from our very experienced resource speakers. Anyone?
Bu Evelyn, I want
Yes.
to ask something to Mrs. Shiva.
Sure.
May I?
Yes, Bu.
Okay.
Please, go ahead.
Referring from presentation, Mrs. Shiva, I would like to ask, I just want to make sure that the current status of the biodiversity subject is not yet integrated with the environmental subject matter in the 2013 curriculum in grade 10 science for schools under the Ministry of Education and Culture. Is it right, or how is the status based on your slide? Thank you.
Terima kasih Ibu. Thank you. Jadi dalam kurikulum pendidikan di Indonesia itu ada sih keanekaragaman hayati. Cuman kalau di SMA jelas ada karena di biologi ada. Tapi kalau di SMK itu jadi tidak detail ada apa tidaknya gitu. adanya di project IPAS saja. Kemudian di project IPAS juga itu belum ada yang namanya modul, belum adanya buku yang memfasilitasi tentang biodiversity and environment. Kemudian yang kita lihat juga sebenarnya lihat dari yang sudah ada modul-modulnya itu enggak, jadi cuman lebih ke memitigasi bencana, tidak ada yang tentang kepedulian tentang lingkungan dan keanekaragaman hayati. Jadi jika memang nanti kedepannya bisa difasilitasi oleh biotrop, mungkin kami akan sangat membantu sekali gitu. Ya, itu. Ada Ibu. Untuk di SMK mungkin yang belum ada, ya. Sama-sama Ibu.
Ms. Evelyn, maybe I will say something about how to measure without the measurement tip or measurement tools.
Okay, thank you very much.
Like for example, if you want to measure the trees, the height of trees, ya Bapak Ibu sekalian, kalau mau mengukur tinggi pohon, misalnya tidak punya alat, ada alatnya macam-macam ya. Like using the broom lice, and also like there are some to measure the height of measurement. So we can use also like using the pen, like a pen. So everybody is already try how to measure the height. Bagaimana mengukur tinggi pohon, ya? But we have there are two men. First is using wearing the pen, and also the other is under the trees. So when someone there in the trees, and then we make it like this, make it into horizontal, and then the man is under the trees. Then take a walk until one line with the pen. So you have that man should have counting the steps. How many steps? How many? So like say 1 meter for one step, and then if they already like this is about 10 steps, it means about 10 meters. Yeah, that is practical. Yeah, exercise in the trees. Also, even I also using this thumb. So when using the nail or whole of the thumb, so that this will be, but it should have the calibrate. Yeah, everybody will be different. Yeah, for example, the diameter of the 30 centimeters or 10 centimeter, it's depend on this on the thumb ya. Jadi ada di kukunya atau di seluruh ini. Kita bisa mengukur seperti ini, like this. That is my experience. And also when we are going to measure the diameter, when the trees, the cylindrical of trees is about more than 3 meters, so it is very it's not easy. So we using this there's a one under the trees, and then using handing the measure the diameter tape, and then make it like this, and then first is this one line with the stem or diameter, and also the other is like this. And then so okay, it's like this. It's the same. It is that is the diameter. So that is there are trees up. And here's an example. So here is surrounding the biotrop. There is trees with the aromatic fragrance. You know, this is melaleuca kayu putih. That is melaleuca kayu putih. This is for fragrance. Yeah. So we can use this is for the medicinal also, like essential oil. So I don't know. In the Philippines also maybe using this melaleuca kayu putih for medicine. This is ada yang tahu Bapak Ibu sekalian ini ya daun kayu putih. Jadi ini wangi sekali ya kalau misalkan kita pakai. Ada di sekitar sini, biotrop banyak. Oke, thank you Mr. Elvin.
Okay, thank you, Mr. Eddie. So, sorry, but our time for Q&A is up. So, for those questions that were not answered on the chat box, may we encourage the speakers to please answer those questions through the chat box, or you can email them if you really want to communicate with them. Based on their presentation, they have given their email addresses. So, to recap, yes, indeed, biodiversity learning by utilizing the school environment can motivate the students to learn and have fun as they undergo the learning activities. Learning becomes more meaningful as the students deal directly with real situations. Yes, it is also very important that the concept of saving biodiversity is already inculcated in the young minds of school children so that when they grow up, they will become advocates or campaigners or champions of biodiversity conservation. Yes, we do agree that biodiversity is important, very important to humans because biodiversity is life. Whatever damages biodiversity also damages the human existence. Hence, to protect and save them, we need to know them, we need to identify them, we need to conduct surveys to monitor their existence, their extent, how much is left, how much is gone to extinction. For we are only encouraged, if not forced, to protect what we know and understand as very vital to our human existence. We do not care about anything that we do not know about that is valuable. So we have to know that biodiversity is important so that we should care about biodiversity. Therefore, we need to know and learn also the proper techniques in identifying and in assessing the development of our flora and fauna, especially the indigenous and the endemic species, those found only in Indonesia or in tropical countries, because mostly of these endemic species are threatened. And in the as mentioned a while ago, we have many endangered tropical species, and some of them are already classified as extinct in the wild and really extinct everywhere. They cannot be found anywhere anymore. Okay, so thank you very much, our distinguished expert speakers, for sharing those valuable information with us. Indeed, these are necessary for Biotrope to achieve its vision to become a reputable center in enriching and promoting the real values of tropical biology in Southeast Asia and beyond. If there were misunderstandings or omissions or discrepancies in the facts presented, please accept my apologies. So without further delay, I would like to turn over the floor to the MC for the distribution of the virtual certificates. Miss MC?
Okay, thank
floor is
you, Bu
yours,
Eve, for
thank you very
facilitating
much.
this session is very well. Okay, now may we present the certificate of appreciation to the speaker given by Miss Risa Rosita to the speaker as Miss Risa Rosita as a coordinator workshop for to speaker one, Ibu Siva, please.
Oh, sorry, it's not virtual, yeah, because
Yeah.
all our 3 speakers are there. It's
Yes.
only me who's not there.
Oke, sertifikat diberikan kepada Ibu Siva oleh Ibu Risa Rosita. Mungkin agak di tengah. Oke. Oke, untuk presenter kedua,
Pada Bapak dipersilahkan. Oke. Oke, kita beri tepuk tangan kepada speaker. And now may we present the certificate of appreciate the speaker virtually. Please. Certificate given by Dr. Suhamsyah Imran, Senior Biodiversity Director, to Bapak T. Heru dan certificate for moderator given by Dr. Suhamsyah Imran to Dr. Evelyn. Okay. Big applause to our speaker and moderator. Okay, thank you. And now we are going to next session is a coffee break. And yes, coffee. Okay, sorry. Next session is we'll start the fourth session featuring two speakers. This session will be facilitated by Dr. Supriyanto. The fourth session. Okay. Okay, this session is featuring two speakers. The first presenter is Nijma Nurfadilah, and the second presenter, Risa Rosita. The topic, introduction, sampling, data collection, and damage caused by fungi, and the data collection and use of soil benefits microorganism, bacteria or mycorrhiza. And the session is facilitated by Dr. Supriyanto. He is a field scientist, semiobiotrop. And so, Dr. Supriyanto, floor is yours.
Thank you very much, Ms. Sheila. Good morning, ladies and gentlemen. This is my second quiz in this discussion or this workshop. I introduce myself is Supriyanto. I'm as a field scientist. I work at Biodiversity since 1981, and now I become an affiliate scientist. And at the time we worked already on some microorganisms like mycorrhiza and bacteria as well. Today we have the two presenters. First is will be on the topic on introduction, sampling, data collection, and damage caused by fungi. And then the second one that will be presented by Ms. Nijma Nurfadilah. And then the second presentation will be on introduction, sampling, and data collection, and uses of soil beneficial microorganisms like bacteria and mycorrhiza. that will be presented by Ms. Risa Rosita. It is also a biotop scientist. May we have the CV of this? Do we have this CV of this our presenters? Yep. This is myself. Yeah. Okay. As you know already. And then the second one is our speakers, Miss Nijma. Yep. This is her profiles. She's working at Xiaomiu Biotopes in 2012 as a member of the research team in phytopathology laboratory Xiaomiu Biotopes, and she has some experiences on post-harvest technology, especially, and she got also master's degree from the same university. Yeah, and now she's working and especially on laboratory as a research team on phytopathology as well. And she has also some experiences as a practical assistant on the topic of molecular genetics, and also in some aspect on the basic of biology. And then the second speakers, my left side, Ms. Risa Rosita. She got her bachelor's from Bogor Agriculture University and the master on soil biotechnology, environmental in the same university. And now she has some experiences on the training in exploration, identification, and multiplication as arbuscular microorganism, fungi, with some companies and also other institutions. And also supervising some students coming from others universities. Now, I would like to invite first speakers will be Ms. Nijma. Time is yours.
Can I share my screen?
Yes, we can see it.
Thank you very much, Mr. Dr. Supriyanto, for the opportunity. Okay, good morning, ladies and gentlemen. Today, I would like to share entitled Introduction, Sampling, and Data Collection, and also Damage Caused by Fungi. The characteristic of fungi, for the first, without chlorophyll, and then the fungi is heterotroph organism and also eukaryotic, and they have cell wall consists of chitin, cellulose, and also glucan. And the somatic structure with hyphae or mycelia, and then the asexual and sexual spores. And the fungi absorb some nutrients from the substrate. Okay, this is the kingdom of fungi. They are 4 phylum. For the first is phylum Cytridiomycota, and then the second is Zygomycota, and then Ascomycota, and the last is Basidiomycota. And this is the example for the species of fungi based on the phylum. Okay, this is the sexual reproduction of fungi. For the first, we can find the plasmogamy and then the karyogamy and also the meiosis. While the asexual reproduction, we can see the fragmentation and then the cell division and also budding and sporulation. And this is the terms of fungi. If we said about the fungi, we can find three terms. For the first is mold, and then mushroom, and also yeast. For the mushroom, for the molds are microscopic fungi, while the mushroom are macroscopic fungi, are included into basidiomycetes, and they have big fruit body. And then the yeast are microscopic fungi, are included into the ascomycetes, and they are unicellular and also has budding cells. Okay, sampling. Sampling is a certain procedure that is followed if a substance, material, or product is taken for the purpose of testing a representative sample of the whole. It is based on the ISO in 2005. And this is all about that we should be considered. For the first, we should know the objective of determination or our testing, and then the sampling officer, and then customer and laboratory. And also we should know the material characteristic and the location, time, and also sampling. And we should know the total sample and also sampling method, and then packaging and transportation, requirement for sampling, and choosing of sampling method. And not only that, we should know the time and budget for sampling also, and then the testing of sample, and the requirement of legal formal and international agreement for observation. For example, if we have this phytosanitary certificate, we should conduct the agreement for the sample. And then the requirement for documentation, and the last is quality assurance aspect. And as we know, the scope of solid sampling is materials in the form of solids, and the solids either packaged or in bulk that has been packaged in the small package. And based on the types, solids can be classified into the two types. They are bulk materials and non-bulk materials. For the bulk materials, we can find the bulk solids and bulk fluid, while the non-bulk materials, such as sheet and then flax and then lumps, and also fibers or yarn. And this is sampling for determining of post-harvest fungi in food and feedstuff. From this figure, we can see they are two kind of sample. One sample in bulk and one sample in package. If we have some lots and we would like to obtain the primer sample, we should take from these three area from the lots, and then we can obtain also the mixed sample from the primer sample. And after we obtain the mixed sample, we can get the subsample, so we can go to the laboratory for the analysis. And this is two types of sphere or probes that can be used for the obtaining of sample. For example, mice or grains and etc. And this is the guideline for checking solid samples. So if we have 1,000 crates, we should divide in the same lots, and we should obtain from the square roots of the total bags by randomly, and the maximum sample is 30 bags. And then if we have lower than 1,000 crates, we should know the sample based on this table. This is based on the Indonesian National Standard in 1998. This is total sample for packaged products in the bags. And then this is table for the total sample for small unit products. So after we obtain the sample from the lots, we can obtain the primer sample. And for the primer sample, we can see the refers to the table. And then this is for total samples in secondary package. Secondary package is similar with the secondary sample. So we can get the working sample after we obtain the secondary package. Okay, this is based on the Indonesian National Standard in 1998. Okay. According to Food Agriculture Organization, if we have 1 until 10 bags, we should obtain all samples. But if we have only the samples between 11 until 100 bags, we should obtain 10 bags by randomly. And if we have more than 100 bags, we should calculate by formulation the square root, the total bags. Okay, this is a guideline for sampling in the feedstuff products based on the Association of Official Agricultural Chemists. Okay, now we go to the preparation to get working sample. We usually use the box divider to divide our sample. And if we have 1,000 grams of samples, we should divide into 2 parts. So we have 500 grams per sample, and then after we have 500 grams per sample, we should divide again into the 250 grams of samples. So we can get 4 parts of sample. And the sample E and C, this is sample for determination of damaged kernels and moisture content, while the B and D are sample for the determination of fungi. Yeah, this is a box divider equipment for obtaining the sample. Okay, this is sampling for mushroom. From this figure, we can see the first stage is cutting off all parts from the mushroom using knife. And then after we cut off all parts of the mushroom, we should make some notes, for example, the date of collection, and also location, and then substrate, grow habit, and we should make sure that the specimen is still fresh and complete. And after that, we should wrap this specimen using newspaper or maybe other paper. And then after we wrap the specimen using paper, we can identify the specimen to the expertise. And then this is the stages for the data collection, especially for molds and yeast. So the first stage for the collecting of data, we should isolate the fungi, and we should enumerate fungi and also identify. And then we can collect the data of fungi. And after that, we should preserve the fungi and create the database. Okay, this is all things that should be considered related on the fungal isolation. For the first, we should know the kind of isolation media, and then the incubation time, and also incubation temperature, and then the objective of sample determination or sample testing, and also kind of sample or substrate. And we should know how we can prepare the sample so we can obtain the working sample by well, and then staff skills or competencies. Okay, for the fungal isolation, there are two types for the methods. For the first is direct plating, and the second is the dilution plating method, followed by pour plate. Okay, for the direct plating method, are to isolate fungi from foods in the form of grains or kernels, such as cereals, maize, nuts, beans, and others. And also to determine the percentage of grains or kernels infected by each fungal species or certain fungal species. And this is some stages for the direct plating method. For the first, disinfect grains. Disinfect grains using 1% of sodium hypochlorite, and then we dry using sterile filter papers, and then we place on the suitable media, depend on the objective of the research. Okay, from this figure we can see this is 1 kernel in the center and 9 kernels in around of the center. Okay, this is a dilution method. Dilution method to isolate fungi from particle foods and also to determine fungal population, so we can get the information related on the total fungal population and also population of each fungal species, colony forming unit per gram sample. Okay, this is preparation. For the first grinding of grains, we use the grinder, and then so we can get the particles like that. And after we obtain the particles, we should conduct the preparation of suspension using water distillation or 0.1% peptone. And this is serial dilution. And after we conduct the serial dilution, we should conduct the plating. The types of the plating method, there are 2 types. For the first is spread plate, and the second is pour plate. But we usually use the pour plate method because it is more homogenized. So we conduct after serial dilution. We use this pour plate method. And after we conduct the pour plate method, we incubate this petri dish and at the temperature room for 7 days. And this is the petri dish. The petri dish should be stored upright. And this is about isolation media. We should know the general media and also selective and differential media. For the general media, we use the dichlorine 18% glycerol agar or the DG18. This media is for isolating xerophilic fungi, while the selective and differential media, for example, Aspergillus flavus and Parasiticus agar, for detecting and enumerating the aflatoxigenic fungi. For example, the Aspergillus flavus, and then Parasiticus and Aspergillus nomeus. And this is for the enumeration. This is fungal enumeration for direct plating methods. From this table, we can see species A. Species A, the color on the surface is black and the bottom is creamish. And we can calculate how many the black fungi in this petri dish. So, after we calculate the species A, we can use this formulation. The percentage of kernels infected by specific fungi is can be resulted from the number of kernels infected by specific fungi divided based on the total kernels in the petri dish. So we can get the percentage kernels infected by fungi. Okay, this is fungal enumeration for dilution method followed by pour plate method. This is species B. On the top surface, we can see this is green color, and the bottom is yellow creamish. So, and the formulation is PCA. PCA is fungal population, for example, species A or species B, and GKCA is number of fungal colonies, and VP is dilution factor. After we enumerate the fungi, we should identify the fungi. So we should know the three kind of identification media. For the first, sulfuric yeast extract agar, and then malt extract agar, and also 25% glycerol nitrate agar. Okay, this is for this, the fungi. This is Aspergillus ochraceus in the three kind of identification media, and this is key identification. Okay, this is references for fungal identification. Okay, after we identify the fungi, we should collect some data. For example, this is for the dilution method. We can make the table like that. So after we get some data, we can preserve the fungi. We usually use potato dextrose agar. And then after 7 days incubation, we use paraffin oil for preservation. And this is database creation in our laboratory. Okay, this is the stages for the data collection for mushroom. For the first, we should collect the mushroom and also making of some notes related on the specimen, and then we should submit the specimen to identify by an expert, and then we should preserve the mushroom and then making of the mushroom database. And this is damage caused by fungi. This is Syncytium endobioticum, can cause the potato wart disease, and it is classified into the phylum Mycota. And this is, we can see from this figure, this is hand that has been infected by Canihamella berthollitii, or we can call this black fungi disease. And this is the micrograph of the Canihamella berthollitii. Okay, this is some fungi that is classified into the phylum Ascomycota. From this figure, we can see this is nutmeg were infected by fungi, Urasium cephalieri, and then this is Aspergillus flavus, and this is Aspergillus niger, and this is Penicillium citrinum. And how about the damage caused by fungi in foodstuff? For the first, automatically the fungi can decrease the germinability and also cause the discoloration of the foodstuff. And not only that, the fungi can decrease the physical quality and nutritional content. And the fungi also can cause the heating of foodstuff, and some of some of fungi can produce the mycotoxins. And this is relationship between fungal infection, mycotoxin contamination, and human health. For example, in cocoa beans, from this figure, we can see this is fungal isolation in cocoa beans. If the fungi can produce the mycotoxins and then contain in the foodstuff, for example, in cocoa beans or chocolate products, or maybe in other grains, it may cause the decreasing of the human health, and not only human health, but also animal health. Okay, this is some disease and kind of mycotoxins and mycotoxin producing fungi. For the aflatoxin B1, B2 can be produced by Aspergillus flavus. Then the disease is liver cancer, and there is so many substrate. And the most dangerous toxin is aflatoxin B1. And this is damage caused by fungi in phylum Basidiomycota. This is, for example, Amanita phalloides. It is very toxigenic because the fungi can produce the amatoxin. The amatoxin can can damage our necrotic and also dermatitis. And also if we eat this fungi, we can die. And this is Amanita muscaria. It is also a toxigenic fungi because it can decrease our digestive process. And if we eat this fungi, we can also die. Okay, thank you very much for your attention. And now I would like to give the time again to moderator. Thank you.
Thank you very much, Ms. Nisma. Please give a big applause for her. Yeah. Thank you very much. So, yeah, we heard about the importance of the real value of the tropical biodiversity. Among others is on the fungi. The kingdom of the fungi consists of mushroom, mold, and yeast. Some of them are useful for human being, but some of them are also harmful because producing a toxical compounds. And when it is eaten by us, could give the effect or negative effect to the human health. Say, for example, about Aspergillus flavus, which is a part of the biodiversity of fungi that could cause the damage on the food stored product, but later on also this damage could produce an aflatoxin that maybe could happen in the food storage, or maybe during the harvesting or post-harvesting handling especially. The effect of this fungal attack could produce a toxical compounds such as aflatoxin that may play important role give the effect on the human health. Therefore, methods or sampling techniques, data collection, and analysis are needed. She show us how to collect the sample from the bulky sample or individual sample, and then how to propagate to confirm the toxical compounds produced by those fungi. Sampling technique becomes very important because it concerns with the food stored product. Yeah. Then if we could conduct the sampling method properly, we will have the data which is reliable, can be validated, all the information, and easily to be analyzed. But there is some other question, of course. some mushroom are edible, but some mushroom which is very beautiful, and then the name is Amanita. Yeah, the name is very nice, Amanita muscaria, because it contains muscarine or cyanide. Yeah, so it is also a toxical component for the human being. Okay, this is my summaries about the first presenter. And of course, there will be another question for the first presenter. Maybe you keep right, and then you can give the answer question later on. And I gave one question before that. How much the aflatoxin in satay sauce? Did you conduct the research? Yeah, because some of the satay producers or gado-gado producer, they don't care about the quality of the peanuts. Yeah, recently there is someone who has died, yeah, because he is also the sauce producer. And the last research, not a research, but the last confirmation from the doctor, medical doctors, he got the heart cancers, yeah, from those one. So it is a homework, yeah, for you. Maybe you please take a look about the satay sauce. Okay. So we will happy for having the second presenters, Ms. Risa Rosita, that she will present the title, Introduction, Sampling, Data Collection, and Use of Soil Beneficial Microorganisms, especially on bacteria and mycorrhiza. Please, Ms. Risa.
All right. Thank you to our moderator, Dr. Supriyanto, for the opportunity for me to present my title. I apologize if I change the title because I don't have much time. If I have to explain about the bacteria and also the mycorrhiza. So my title is Introduction, Sampling, Data Collection, and Uses of Soil Beneficial Microorganisms, AG and PB Reducing Bacteria. Here we call it AG as a mercury and PB is a lead. By me, Risa Rosita. I am a head section of Environmental Technology and Security ETS of Siameo Biotrop. So at current, we have one program as a Biotrop, one of Biotrop priority program. It called as Safe Biodiversity for Future Generation. So our program is want to emphasize the need to carry out the transfer of knowledge on biodiversity conservation as early as in the young minds of high school or vocational school students in Indonesia and Southeast Asia. We know that a school is a place where children gather to gain their knowledge, and this knowledge transfer is one of the primary roles of the teacher as an educator. So, in accordance with Siameo Biotrop vision, it's necessary to inculcate a deeper understanding of the value of biodiversity starting from the teachers or educators. Hence, Biotrop will conduct a study on the biodiversity awareness among the high school and vocational school teachers in Indonesia and Southeast Asia. But referring to Mrs. Siva presentation, so we will study only focus with the vocational high school in Indonesia. Now we are looking for the potential partners from Indonesia and also from Southeast Asia. Based on Wilcox, biodiversity is Sorry, my signal is not working properly, so my slide couldn't move. All right.
Okay, we are waiting for the slide presentation. But anyway, Ms. Rosita is still preparing. But for having a good discussion later on, we may know that some organism, especially soil microorganism, is very important, as stated yesterday by Professor Kitaru from Japan. Microorganisms, especially mycorrhiza and also the bacteria, plays important role in our planetary boundary. This was very nice yesterday's discussion. Planetary boundary, it become our important things, and especially on the nitrogen, phosphor, and others nutrient which is important for the plant health as well as for plant growth. Yeah. And now that we have also the presentation on the microorganism, especially mycorrhiza. Mycorrhiza is a very good for promoting the growth of the plant, especially when we are planted in the very difficult condition or very critical condition. And we may know that some bacteria, such as rhizobium, plays important role in capturing the nitrogen from the air. Yeah. And we can develop the technology on the nitrogen fixing bacteria as well, and also mycorrhiza. I think you are ready, Ms. Rosita. Okay, time is yours, please.
Thank you to moderator. All right, today I would like to present my title is Introduction, Sampling Data Collection and Uses of Soil Beneficial Microorganism, AG and PB Reducing Bacteria. AG is mercury and PB is leads by me, Risa Rosita from Sayameo Biotrop. Next slides, please. At current, ETS section has one program. We call it as a Safe Biodiversity for Future Generation. This program is to emphasize the need to carry out the transfer of knowledge on biodiversity conservation as early as in the young minds of high school or vocational school students in Indonesia and Southeast Asia. We know that school is a place where children gather to gain their knowledge, and this knowledge transfer is one of the primary roles of the teacher as an educator. So, in accordance with Sayameo Biotrop's vision, it's necessary to inculcate a deeper understanding of the value of biodiversity, starting from the teacher or educators. Hence, Biotrop will conduct a study on the biodiversity awareness among the high school and vocational schools teachers. Based on Mrs. Siva presentation before, we will focus on our study in vocational high schools, and now we are still looking for our potential partners from Indonesia and also from Southeast Asia. Next slide, please. Okay, based on Wilcox, he said that biodiversity is the diversity of living things in all biological levels, from nature to biomes and ecosystems. And based on the United Nations, they said that biodiversity is the variability of living things, both terrestrial and marine, as well as the variability within species, between species, and ecosystems. Based on Gaston and Spicer, biodiversity is a variety of all forms of life in a site. So how is the environment? Based on Amshari 1989, it consists from two aspects. First is physical environment. It's everything that's around humans. The form is inanimate objects such as air, stones, houses, light, and so on. And for the biological environmental aspect, is everything that is around humans. The forms is living things such as animals, plants, and microorganisms. Next slide, please. Okay, this is advantage of biodiversity for the environment. Is for environmental service, the positive impact on the environment given by flora, fauna, and microorganisms in a biome. These positive impacts include first, maintaining the stability of the water cycle, promoting the formation and protection of soil and soil organisms, storing and recycling nutrients in the soil, reducing and absorbing pollution, maintaining stability and influencing the local climate, maintaining the stability of the surrounding environmental ecosystem. So therefore, it's very important to maintain the biodiversity of an area. Next slide, please. So here I would like to share our study about biodiversity and environment entitled Isolation and Characterization of AG and PB reducing bacteria in several contaminated habitats. Next slide, please. Some of developing countries has some issues, such as industrial growth and increased use of fuel oil. It causes pollution by heavy metals. And the second one is mining, metal smelting, industrial agriculture activities, and domestic activities cause heavy metal contamination. So heavy metal is so dangerous, toxic, can be degraded, can be destroyed, and persistent. So heavy metal is one of the major threats to aquatic fauna, especially fish, which are one of the main sources of our protein-rich food for humans. So one of methods to reduce heavy metal contamination is using bioremediation by involving AG and PB reducing bacteria. Next slide, please. So our study aims is to isolate AG, mercury, and PB reducing bacteria from several contaminated habitats, to characterize AG and PB reducing bacteria, and to measure the effectiveness of bioremediation. Next slide, please. So to collect the data in the methods, we're doing 4 stages. First is sampling. We took 2 samples, solid sample and the liquid samples. For the solid samples, consists from a brick factory soil, PBTK, and then we took from Bogor Botanical Gardens river sediments. The code is PKRB, Pongkor X mining soil, PPKR. And for the liquid sampling, we took the sample from Biotrop liquid waste, CBIO. After taking the samples, we did for the isolation by using spray plate method, and afterwards we have to characterize for the morphology, physiology, and biochemically. For the morphology, we did the microscopic observation, and for the physiology, we did a hypersensitive test, gram staining, catalase test, oxidase, and motility test. And the last one is biochemically, the bacteria growth test at various pH, and we did the various ppm tests. So how to measure the effectiveness? We measure of the ability of heavy metal reduction by using spectrophotometer, bioassay test, and data analysis. We using for the SAS9 program, analyze for foreign and food testing with the Duncan test at an alpha 5% level. Next slide, please. So here is our sampling location at Bogor Botanical Garden River, Pongkor X mining soil. Next slide, please. Biotop liquid waste and brick factory soil. Next slide, please. Up. Okay. This is for our isolation stage. Four samples consist of PBTK, PKRB, PPKR, and CBio. 10 grams of the samples had been weighed and transferred into 90 milliliter physiological solution, 0.85% NaCl per sample. After that, we homogenize the samples 30 minutes, and we did for the serial dilution from 1 serial up to 10. After that, we transfer 0.1 milliliter to LB media, Luria bertani. This is selective media that we already used for this study. The pH is 7.2 until 7.4 with the spray plate method. And then after that, we incubate 5 days at room temperature, and we did the characterization test for the morphology, physiology, and biochemically the bacteria. Next slide, please. So here is the result of the isolation. Next slide, please. Yeah. After doing the isolation, we can collect the data. From the result, we obtained for the 28 bacterial isolate had been isolated, consists of 17 isolates are PB-reducing bacteria, and 11 isolate suspected as AG-reducing bacteria. The bacteria population was successful growth and serial dilution, 5 dilution up to 10 from CBIO samples, henceforth from PBTK, PKRB, and PPKR sample. Next slide, please. So here is next characterization steps. We did a hemolysis test and the hypersensitive test. From the result, we obtained for the 17 pathogenic bacteria, consists of 11 non-pathogenic bacteria, and non-pathogenic bacteria were 2 bacteria took to be able to reduce AG, and non-bacteria took to be able to reduce PB. Next slide, please. Here is a result from the Oxidase Test. 3 bacteria isolate, PBKRB1, PBBio7, AGKRB1, show positive reaction on the test. The positive reaction was indicated by the appearance of a purple color on the oxidase paper. Next slide, please. And this is for the Catalase test. The isolate PBTQ1, PBTQ5, and PBKRB1 produce more catalase enzyme. Some pathogenic bacteria will produce catalase to defend themselves against hydrogen peroxide attack. It's a weapon commonly used by the host immune system. Next slide, please. So here is for the motility test, the result, all of the bacteria isolate are motile. The positive reaction indicated by the distribution of colonies when grown on sulfide endomotility semi-solid media. Next slide, please. Yeah. Okay, here is our data from the characterization, morphology, psychology, and biochemically. All 11 isolate had a cocoa cell shape, generally gram-positive. Most of the colony shape are irregular, flat, entire, and the surface is dull and smooth. Next slide, please. Up. Please, up. Okay, this table talk about mortality of golden fish, Cyprus carpio, after 24 hours treatment. So there is a very significant difference between the treatment of concentration of metal contamination Ag and Pb with the number of death of golden fish, Cyprinus carpio, more than 0.05. And AGKRB5 was capable to remediate 1 ppm no mortality and 10 ppm Ag contaminated water after 2 hours of inoculation. Next slide, please. The conclusion of our study, 2 potential isolates were successfully isolated, consisting of Ag reducing bacteria, AGKRB1, which was isolated from sediment samples from the river of Bogor Botanical Gardens, and PB reducing bacteria, PBBTK1, isolated from soil samples from the brick factory. AGKRB1 isolate was able to synthesize both catalase and oxidase enzyme, motil and alkalophilic. Meanwhile, PBBTQ1 isolate can synthesize catalase enzyme, motil, and growth at various pH. So, in this study, the effectiveness of bioremediation of AGKRB5 was 99% with the mortality 67%, and PBBTQ1 reached 100% no mortality there. The result of the bioassay test show AGKRB1 was capable to remediate 1 ppm and 10 ppm of Ag contaminated water after 2 hours of inoculation. Next slide, please. So, after we doing the combined study of the biodiversity and also the environmental, so we can answer from the study the positive impact of the environment given by flora, by microorganisms in a biome. These positive impacts include first, maintaining the stability of the water cycle, reducing and absorbing pollution, and the last one is maintaining the stability of surrounding environmental ecosystem. So that's why here we all are gathering to arrange how to inclusion about the biodiversity subject into the environmental education curriculum. That's all from me. Thank you.
Thank you very much, Ms. Risa. We give big applause. It's a very nice presentation, how to introduce the microorganism, especially bacteria, which is beneficial for the environment, for the environmental stability. So soil beneficial organisms like bacteria And mycorrhiza, for example, plays an important role in biodiversity and environmental conservation, and among others in reducing the pollution effect of mercury, erarchium, and lead or PB for maintaining the environmental stability through bioremediation technology. In this case, the role of microorganism in maintaining the environmental stability was proven, this among others, by bacteria, say for example by Staphylococcus and Streptococcus, in reducing the water contamination from the mercury and lead. And the fact, by introducing this microorganism, could increase the survival rates of golden fish, say for example, in this case. Therefore, it is also important to be introduced by including this kind of beneficial microorganism to the school, to the university, to the students especially, and through the improvement of the curriculum and some part, and by including the most important one, by including in some curriculum like biology, environmentals, etc. Therefore, but still some question maybe in this case, how much the mercury and leads content in the golden fish? It is maybe for the next research, Ms. Risa, yeah, and maybe you cannot answer now also, no problem at all, okay. But we give the opportunities to the audience to give the question. Maybe from the chat room, please check from the chat room. Yeah. Yeah. Okay. So you can raise your hand. Okay. This the question from Ibu Evelyn. Yeah. For the cancer causing by fungi, do they develop cancer after one ingestion or of the contaminated food, or after many intakes of the infected food or food substitute? Thank you. Jadi apakah langsung kena atau mungkin beberapa setelah banyak memakan itu, menumpuk gitulah, mungkin juga bisa jadi. Okay. And then the second question is coming from Pak Yadi Mulyadi, yeah, from Cianjur. Since the biotope, could you please? Okay, yeah. From Pak Mulyadi, maybe from. Okay, could you please? Oh, how many questions? A lot of questions. Okay. I observed that in PrEP, some school in Indonesia, topic of microbiology such as fungi, taxonomy are not updated yet. Okay, this is good. Teacher teach the out of dated knowledge that perhaps has been revised or revisited. Meanwhile, improvement of teaching quality on the school facility, including laboratory, workshop, etc., seem very limited. How do we deal with this kind of problems? Okay, so maybe by inclusion, say for example, don't eat the peanut with the bit taste, something like that, because it contains aflatoxin. It is also one of the education. It is also good, yeah, to know what kind of the say for example peanut containing aflatoxin from morphological point of view. For example, before they eat and die. Okay. For the cancer-causing fungi, do they develop cancer after one ingestion of contaminated food? It is already read, yeah. And then the from Dr. Evelyn, is there a cure from liver or kidney infection caused by mycotoxin. If so, how? Ya, jadi kalau ada yang kena, gimana cara ngatasinya? This for Ibu Miss Nisma. Please, Miss Nisma. Yeah.
Okay, thank you for the question.
2-minute answers, please.
Okay, please. Thank you.
Time runs, yeah.
Okay, thank you, Dr. Evelyn, for the question. There is still very limited the research related on the medicine that can be used to the fungal infection, because some research only for the prevention. So how to prevent the fungal infection in the foodstuff or feedstuff? We should prevent by monitoring the quality of foodstuff or feedstuff from farmers and to consumers to be consumed, and then, for example, for good agricultural handling, and then good post-harvest handling, and also good manufacturing handling, and then the good distribution handling. So there is still very limited the research related on the medicine for fungal infection, but we can prevent for the fungal infection. And then for the question, the second question from Mr. Samir.
Yes, please.
Mr. Samir,
could you
can
you share the
bamboo
question?
fungi?
Yeah. Can you share the
Okay.
question, please? Yeah. The question.
Okay, can now bamboo fungi. I mean the bamboo fungi for the species Palus indiciatus. The Palus indiciatus is edible for our human health, so we can eat the fungi. Because in China, the Palus indiciatus can be used for the soup, and then the morphology of the fungi. The fungi has fruit body with the conical or belt shape. So we can see from the morphology the cap on the fungi. And then the question from
Dr. Evelyn.
Miss Mega.
Oh, okay.
Okay, maybe we can collaborate together by making all the MOU, so we can work together in research maybe or in training. Thank you.
Thank you. So if someone want to be the mushroom hunter, someone want to be the mushroom hunter, one of my suggestion is, before eating the mushroom, if you don't know well about this classification, etc., you just drink the milk first. Yeah, so we'll get the diary maximum. Yeah, it is my experiences when I was a mushroom hunter in France. Yeah, so this is my experiences. Okay, next question for Miss Risa, please. Can you, may I see it? Yeah. So nobody asked her. I will ask her. Okay. So how much the mercury content in the golden fish? Okay. It is very important also. Don't put any waste containing the mercury. Say, for example, battery. Yeah. Battery waste. Don't put any anywhere. You have to put in the proper place because it contains the mercury, and this mercury maybe goes anywhere. It is very important for curing our environment. So maybe we can develop later on about the collection of this microbiology, this microbe, especially the Streptococcus. Streptococcus also is a danger for another diarrhea problem, of course. So of course we have to take care about that. Okay, because no question, I think we have to conclude this meeting, and we will go to the focus group discussion. And we would like to appreciate all the speakers who please give big applause. Thank you very much. So I give the time for the master of ceremony, please. Yeah. Ibu Sela, come in.
Okay, thank you, Dr. Suprianto, for facilitating this session. And now, we present the certificate appreciation to speaker and moderator. The first presenter to Ibu Nijma, given by Manager Human Resources Bapak Bambang. Okay, please. Oke, sertifikat pertama diberikan kepada Mbak Nijma. Sip. Oke, silakan ke sini. Oke, sertifikat pertama diberikan kepada Ibu Nijma Nur Fadila. Kita berikan tepuk tangan, big applause for the speaker. And the next presenter pada Ibu Risa Rosita. Oke, sertifikat diberikan kepada Ibu Risa Rosita oleh manajer Harat Bapak Bambang Sulistyo. Oke, kita berikan big applause. Oke, untuk moderator, silakan Bapak Dr. Suprianto. Sertifikat apresiasi for moderator Dr. Suprianto. Okay, big applause for speaker and moderator. And now, ladies and gentlemen, next session is a break for lunch and pray, and kindly be reminded the next session FGD will be start at 1 p.m. Okay, we will appreciate it if you could return to the room on time. Thank you. biodiversity in the environmental education curriculum. So the next session is FGD, and this session is will be divided into 3 breakout rooms. The moderator for each room are Dr. Supriyanto of Simayo Biotrop, Dr. Sri Suhartiningsih of the University of Kristen Satya Wacana, and Dr. Romi Ardiansyah, the head section of Ecosystem Restoration and Conservation of Simayo Biotrop. Okay, for your information, the focus group discussion topic, the 3 topic. The first topic about the biodiversity study fauna, and the room 2 itu biodiversity study flora, and 3 biodiversity study microorganism. Okay, before continue FGD, I'm please Miss Risa Rosita, can you explain guidelines for FGD? Please.
Okay, thank you to MC Mrs. Sela. So for the all participants, we are proceed to go on the focus group discussion. So here we already has one questionnaire. So each group it should be arranged, revised to make our questionnaire to be clear. So in presentation session, we can get a conclusion about how to create for the biodiversity questionnaire. And our moderator, they will guide us to proceed the focus group discussion for each room. Thank you.
Okay, thank you, Miss. So the participant are now invited to join the breakout room. Can you choose room 1, 2, or 3? And the topic for 1, fauna, 2, flora, and 3, microorganism. Please, the participant join to a breakout room. Oke, untuk peserta yang di ruangan Jati, ini untuk FGD topik pertama, yaitu breakout room 1, itu ada apa? Fauna dengan moderator Bapak Dr. Supriyanto. And the room Kenari itu untuk breakout room 3, Dr. Romi Ardiansyah, topik the microorganism, and the room 2 itu kita di ruang podcast lantai 1 di ruang bawah. Itu dengan moderator Dr. Sri Suhartiningsih. Jadi Bapak Ibu silahkan memilih untuk bisa bergabung di mana terserah sesuai dengan topiknya, apakah mau di room 1 di sini dengan membahas fauna bersama Dr. Supriyanto. Terus kalau yang di room 2 itu membahas flora di podcast, dan di Kenari itu mikroorganisme bersama Pak Dr. Romi Ardiansyah. Oke, silahkan Bapak Ibu. Untuk reporter pada ruang 1 itu ada Miss Wiwi, dan 2 itu ada Oke, saya ulangi. Untuk reporter pada ruang fauna itu ada Deda, dan flora itu ada Mas Haris, dan mikroorganisme ada Mbak Wiwi. Seperti itu. Mbak Deda, Pak Deda. Ada Mas Haris, Bapak. Oh. Deda udah ke bawah ke ruang podcast.
So, so who
Dear all participants, while we are waiting for Fauna group, please fulfill our post-test and also evaluation form. Those links are attached in our chat room and screen. Thank you. Bapak Ibu peserta pelatihan yang ada di ruang Jati, silakan dapat mengambil coffee break. Terima kasih.
Dokter Sri siapa namanya? Oke. Okay, ladies and gentlemen, welcome back to the main room. We hope you had a fruitful discussion and still be with us until the end of the workshop. So I will ask to our moderator to give a discuss report. For the first one, we welcome Dr. Suprianto to present discussion report, the topic of flora. Dr. Suprianto, time is yours.
Thank you very much for presenting the discussion in the room number 1. So I would like to have your favorable services on presenting my reporters, Mr. Haris. This is what we have in our discussion concerning the format for questioning the biodiversity conservation schools, especially. Some of them were modified because maybe quite difficult to be understand about the question sometimes, and sometimes also we have to ask correctly because the quality of the answers depend on the quality of the questions. Okay, so this one about the names of the one who filled the formulas, on filling the form, and it is nobody have the objection about this one. So we continue. About the instruction, sometimes the answer is, sometimes we would like to have the scale from 1 to 4. Why we don't put the 5? Say strongly disagree, agreed, or strongly agreed. So we don't put moderate, because sometimes if we put the moderate, anybody prefer to choose the moderate one. It is yes, it is our condition, something like that. But sometimes also the question must be answered yes or no. So the yes or no question is not is not given yet in the instruction. Okay, and then check one or more question if there is the other choice of statement, and then sometimes also if there is no choice in the choice statement, so we can put the other box to put the answers, or maybe you can add some of them. Okay, next please. Then the part 1 about the biodiversity knowledge. Principally, we divide it in 4 chapters and 4 parts. Say, for example, about this one, the introduction of biodiversity. Do you know the concept of biodiversity? So the answer is yes or not. It is modified accordingly about the question. Next, please. And then about the knowledge on the loss of biodiversity, we have to check about the factor causing the biodiversity loss. So we can also have to put a checkbox if we need. So it can be because of loss of habitat, pollution, waters and soil air pollution, and also climate change. Climate change also could be one of the factors. Seawater heating, ocean acidification, and then the emergence of disease, exploitation of flora and fauna, and then migratory or invasive species, agriculture and forest industrialization, and others. And then the biodiversity has the benefits, yes, but strongly disagree or not, yeah. So this is what we have. The question. Next, please. The benefit in preserving the biodiversity. This coming from the small things that maybe more give the impression about the biodiversity. So it can be improve air and water quality, and then the climate. That was the climate control, but in the school maybe microclimate. That is more sensitive than the climate control in the sense of knowledge. Pest and disease control, we can show also the student about the presence sometimes about the pest and disease in the plant, something like that. And then the pollination, because it related to food production, it is very sensitive to better preserving or understanding about the biodiversity. And also the source of other uses, say for example for drug, herbals, and also medicinal plant. Of course, it is the most commonly in the traditional community or social, and we can have some experiences, say biodiversity conservation is started from the kitchens, because we collect some spicy species, something like that. And sometimes for the medicinal plant. Flood control, and we add disaster prevention and mitigation. And then recreational values, of course later on we will develop to botanical garden and others. Next please. And then the efforts can be for saving the biodiversity, can be conservation of natural resources. You know that we have the, for example, about the protection areas or others, or protection forests, etc. Then habitat restoration. We know that sometimes there's some disaster that must be restored about from landsliding, etc. Prevent water and soil and air pollution, also it is known, yeah. And then to ban the entry of invasive alien species, so must be given as knowledge for saving the biodiversity, promoting the importance of biodiversity in society, using local threatened or endangered species. We add the words of threatened and endangered species. And then also we have to add this one is we add about the enriching the collection of useful plants and herbs, maybe in the school, that is the most pronounced. Okay, next please. The academic discipline that contribute to the shaping of biodiversity, in this case the concept is inclusion only. So that's not really, except if it is the faculty of forestry or faculty of forestry which develop the, of course, about the conservation. Yeah, it is really become a matters or subject of education. But in the school, we just put as an inclusion to some natural and applied science such as biology, chemistry, computer science, engineering, geology, mathematics, physics, and medicine. Say for example about the chemistry, we can introduce them about the different aromatic plant, which is having different function for medicine, etc. And with the computer science, we can develop also architectural analysis using computer science. The engineering, we can introduce why the plant in the beach, say for example, about the coconut, very strong to the wind, say for example. The geology, we can introduce them there because of some geology produce mineral, there are different mineral, or some geological formation give specific mineral to the plant, the area become acid or basic, etc., which also affect to the plant communities, etc. And then in the social science, we can use about the anthropology. We know about the maybe about the use of certain herbs in certain society. Yeah, I forgot the name about this. The term, Pak Romi, maybe do you still remember about the relationship? I forgot the name. Okay, about the specific culture using a specific medicinal plant, something like that. Do you still remember? Ethnobotany, yeah. The name is ethnobotany. So we can put the ethnobotany. And then for the humanities, we can see about the art, history, language, literature, music, and philosophy, religion, and theater. We can introduce the biodiversity in this kind. Business, yeah, accounting, economics, finance, also we can introduce it. And then education of science, there's a lot of things that can be introduced in this case because it's related closely to the anthropology, etc., and arts. Yeah. Next, please. And then this one, we introduce. Oh, next, yeah, this is him. Yeah, and then the second part about the concern of biodiversity. You have any concern on biodiversity? Yeah, strongly concerned, etc. This is the introduction. But the next question is the second question, please. Your country is a country with the high biodiversity. It is increased in the country's level. So maybe we can say it is a different countries, say for example Timor-Leste, because it is hard and hotter, and then maybe some countries are having a desert area, but if we compare to the tropical with the humid tropical forest, maybe yes, a high biodiversity, but we can agree or not agree about that. Okay. And then the second question about the efforts. About the efforts, yeah. Is there any efforts to protect the biodiversity in your country? We modify it accordingly. There is some nature reserve, nature conservation area, and then we add, among others, is about the conservation through breeding. Yeah, sometimes we have. Say for example about in Malaysia there is a breeding on the rhino, yeah, because rhino is become an endangered species. And then botanical garden is also one of the effort from the countries, yeah, to develop to conserve of biodiversity in ex situ condition. And then about the restoration, etc., and then prevent and of the invasive plant species also. And then the important one about the inclusion of the biodiversity and environmental education in curriculum. So if it is an inclusion, so you can modify it accordingly, the method according to the local condition, of course, and then the local resources. Yeah, please, next. And this already, next please. And then the effort that has been made, yeah, it is related to number 3, and then, well, we just agree or not agree, yeah. The effort that has been made, yeah. Sometimes we have to put some box, maybe just only how many percent they have been made available. So maybe in this session we don't put yet the scoring system yet. Yeah, okay. Later on about the scoring system will be made available by the committee. Number 5. The group that is responsible for protecting the biodiversity in your country. Of course, the first one, government, private sector, international organization, college, student, school teachers, yeah, student, lecturers, local community, and NGO as well. So we can put some others, maybe because of our knowledge is limited, so you can put the others. Okay, thank you. Next, please. Now we speak about the efforts. Is it easy or not to promote? And then what we mean is how the efforts can be easily made available. Say, for example, by promoting biodiversity on social media. One of my friends promoting by putting, say, for example, the words biodiversity in my backyard. So every day he posting the photos about the biodiversity in his yard, in the backyard. And sometimes we got there some beautiful flowers, beautiful leaf, beautiful animal, something like that. Of course, biodiversity in the school environment, all local people, and then the volunteer also it is must be considered. Keep the environment clean. Also, this one become a issue. Good issue this morning and this afternoon. And then integrating the biodiversity material into environmental education curriculum in the school. It is pronounced also and become good issues. Yeah. And then developing the educational boards. These new things. Yeah. But in developed countries like Australia, they made it about the board and sign on environmental protection, and there's some question, and in the board there is a question and also there is the answer. So any children can go there and then take a look, what's this animal, and then there's a name, what kind of the food, the feed for them, also there is also. Next please. Part 3, biodiversity and education. Yeah. Environment, so are you agree or not to satisfy? How the material be provided and the knowledge was delivered by the teachers. Okay, then yeah, are you satisfied or not? So we can put that. Okay, next please. And then this is, you satisfied or not with the material? Yeah, we can answer. Where did you learn about the biodiversity? Maybe formal education, maybe through training, maybe through surrounding environment, maybe from the book or magazine, and also article, news on internet, maybe family, yeah, journal, etc. Storytelling can be also. Next, please. The phenomena that raise your awareness of the importance of the biodiversity. Sometimes shocking things become or increase our awareness. Say, for example, suddenly the absence of the fish from the river. Also, this shocking. Yeah? So become aware. Then every day maybe they put one fish in the river or in the river bank or whatever. Become the awareness of this people is increasing. Lack of insect and other wildlife can be. And then homogeneous forest, monoculture forest, or maybe harvest failure due to climate disturbances or climate change. And then diversity of animal and the zoo, because sometimes, okay, finally someone knows about the biodiversities in the zoo, because it is not coming from the Indonesia, for example. Yeah, we can see in the zoo some diversity of the animals. And then natural disasters like tsunami, landsliding, forest fire, heat wave, flood, tornadoes, climate change, etc. Also become sometimes someone become aware to the biodiversity, the role of biodiversity. Next, please. Biodiversity curriculum is essential to be taught in the school. Yes or not? Yeah, agree or disagree. If yes, then what kind of the curriculum? The question number 6. Yeah. Oh, number 7. Sorry, please. Number 7. Yeah, the level. Yeah, can be in kindergarten, but in different ways. Yeah, because kindergarten just know colors, smell, something like that. Yeah. So we can modify it. Say for example about the color of different leaf. Yeah. Shape of different leaf, it can be part of the education system. And there may be different smells of the medicinal plant, or maybe different flowers or different leaf could be used for introducing the biodiversity in kindergarten. Of course, in the elementary and then junior high school, and then also high school, senior high school, can be done accordingly, can be modified accordingly, depending on the situation. Next, please. The level, yeah, we put here formal and non-formal education. We put the boarding school, say for example, it is also important. Next, please. Yep. Number 9, factor causing lack of understanding. So why someone doesn't know about this biodiversity? Yeah, all insufficient or lack of. Yeah, insufficient lesson, insufficient resource, incompetent teachers, insufficient learning media, or lack of student interest. Yeah, can be like this. Okay, next. Then number 10, the most preferred learning. Yeah, can be for studying biodiversity, can be on-site, online platform, practical, electronic learning modules, video games, magazine, or storytelling. This is we add the storytelling. Yeah, for the kindergarten, most of them prefer storytelling. Sometime in the elementary school as well. Next, please. Part 4, the role of the SDGs. It is introduced. Yeah. For the SDGs, there is some 17. Do you know, or maybe do you understand about the 17 development goals? Yes or no? Yeah. Yeah, okay. The next is, if yes, say for example, what we have to do? Number 2, please. Question number 2. Agree or not agree by preserving the biodiversity? Yeah, next please. And then which goal concerned with the biodiversity? Of course, this is not related closely with the elementary school, etc. Even, of course, kindergarten is not so related. Maybe this is for the better, the higher education. Next, please. Yep. The concept of 17 goals of SDGs has been taught in the school, in curriculum. Yeah, can be yes, can be not. Yeah, agree or not agree. Okay, next please. But there should be introduced because the number 4 is the quality of education, and then the biodiversity may be related closely with the quality of education. And then about the this one, the level, yeah, can be modified accordingly. Of course, depend on the levels of the education. Next, please. That's finished? Okay, so this all. If you would like to have another edition, please chat in the box, yeah, and maybe you can send directly to the organizing committee for our consideration. Thank you very much for your kind participation, and big applause for us, to all of you. Thank you.
Okay, thank you, sir, for the report discussion. And next, we welcome Dr. Sri Suhartiningsih from University of Kristen Satya Wacana. to present, discuss, report the topic microorganism. Dr. Sri Suhartiningsih, time is yours. Oke, masih mute. Oke, please, Dr. Sri Suhartiningsih.
Yes. Okay. Thank you, participants. I present about the results from FGD's microorganism group, and I share PPT from, and then questionnaire. Yes. Okay. The result from the FGD microorganism. Overall, the questionnaire can be accepted by the participant. Yes, there are several input of substantive questionnaire. Number one, teachers and students should pay more attention about flora, fauna, and microorganisms. And biodiversity or kindergarten. For studying biodiversity are not from elementary school, but from kindergarten. And about climate change, not only we talk about the climate change, but also climate amelioration. This is arguments for like of is needed so that teachers and students can have the same understanding of business that care about biodiversity. The example of calculating economic costs that include aspect of environmental protection. This is a resume for our discussion. And then the questionnaire, we review the questionnaire. Yes. Start of the part of one, and number 5, the benefits of preserving biodiversity, but not climate control, and we just climate amelioration. Yeah. And number 7, efforts that can be made provide more meaningful learning to students. And number 7, the academics disciplines that can contribute to saving biodiversity, sports, physical, and slides. Yes, part 3, biodiversity, but not yes, result you can access to studying biodiversity formal education, kindergarten, elementary school, middle, high school, and university. And number 7, the levels of formal education that is important to teach the concept of biodiversity, as we agree the question and answer. And number 9, concept of biodiversity. We correction the statement, lacks of knowledge of the teacher, lacks of concern from student on the importance of biodiversity, lacks of concern from teachers on the importance of biodiversity. And part of 4, this is we things agree the questionnaire. I think the finish we achieved from microorganism group. Thank you.
Okay, thank you, Miss Dr. Siswatiningsih. So the last report from Dr. Romi Ardiansyah. Topic is fauna. Dr. Romi, time is yours.
Oh, okay. So, oops. No, my face is too big there. So, thank you very much for the opportunities for me to be the moderator for today's afternoon FGDs. But first, I'm sorry because it takes too long, and also it's not so really finished actually. But yeah, I will start. For maybe you would know it's not so really finished because I will start to about the form, about the questionnaire. So okay, I will make it big. So there are several questions, several points also that we have discussed this afternoon, and there are also several changes. So that we have to change to make it clear and then to make it also easy to understand for our respondents. So maybe I will start from here. This for the name, school, country. So for the country, we would suggest to change it's not a country but to be the nationality, because country nationality maybe is that's also similar, but it's normally we use the term is nationality, not the country. So that's our input for this kind of the biodata. This kind of the questions, and the others is fine. And for the instruction, first we discussed about this. We somehow quite confused because there are two types of the questions. Because they're confused about the strongly disagree until agree. This kind actually we changed this. Not from negative to positive, but from positive to negative. So we change it from the strongly agree until strongly disagree for the both. So that's we changed, and this, but I didn't change it here, but I changed it in the questions. And the second one, they put a check mark on one or more options maybe. So because there are several options there, so we have to change one or more options. So we change to be option, not the questions, because there's not really questions. It's just the options that we have to choose. That's what we changed. And the second, we changed this actually. So it's not the concept of biodiversity, but we change it to be the definition of biodiversity. So do they know about the definition of biodiversity? But so I said before, so we changed from strongly agree until strongly disagree. So we make it from positive to negative. So that's what we need to change the concept to be definitions. And the other, also the biodiversity, this is fine. We have also, and we have several this some kind of questions. Do we think about the environmental affect biodiversity or biodiversity affect the environment? Because it would affect these questions actually. So that's what Dr. Supriyanto said. It depends on the questions. So that's, and we also have, we would like to add the questions because this is for the education for the students. So they would aware about that human activities, that their activities are could cause the biodiversity loss. So do they agree or not about that? And we also put your life depends on biodiversity or your life depends on environment. So there are three questions that we would like to add in this questionnaire. And then about the biodiversity has benefits. So our question is benefits to what? So our in our team and our focus group discussion, we try to make it clear. Oh, this biodiversity has benefits to environment and human being. Because the first thing we put it only human being, but we think okay, maybe we also put the environment because we would like to add, because this biodiversity should have benefits on environment also, but also would benefit in human beings. So not only benefit in human being, but also in environment. And this one factor that can cause biodiversity loss. So for this, we add several, but we also remove several. So we add about the habitat fragmentations, and also we also add about the fire forest, and also is this some kind we not really add, but we refresh it to be the introduction of alien and invasive species, but not only the migratory or invasive species, and also we put the conventional agriculture and forest industrialization, but we omit or we remove three answer like seawater heating, ocean acidification, and emergence of disease, because it's somehow the effect of the climate change. So this climate change could already take everything in the above. So we suggest to omit these three questions. We remove these three questions, and this is one for the benefit of preserving biodiversity. We would question. So in our group would question climate control. How do we control the climate? That's the question actually. So that's the question mark. So we would like also to know what is the meaning of the climate control, and we also change the drugs to be the medicine. Maybe medicine is really the right term about the sorts of the medicines. And this one effort that can be made to save biodiversity, it's quite long actually to discuss this one, because the first we have the conservation of natural resources, which is also have related to the enhancements, restoration, and maintenance of the wildlife and habitat. So we add this, we refresh this question, these options. This still, prevent water, soil, and air pollution, yes. Ban the entry of alien and invasive species. So we put alien species in there. And we refresh the promoting the importance of biodiversity in society to conduct the informations, educations, and communication campaigns on biodiversity. So that's somehow we change it. But however, this kind of answer actually, like the effect of, if we do this conservation of natural resources, so we got this 4. So what to do about this 4? Is it like this is general, after said like from general, so we just take, okay, just put the general answer there, we also put the specific answer there. However, they actually have connected each other. That's also, and this is somehow make us like wondering why this question we put in here, like academic disciplines that can contribute in saving biodiversity. So we make we somehow like we would like to change this, or not change, like to change the sentence to be, in what subject you learn about biodiversity. So because the point of this workshop, the point of this questionnaire was actually to include inclusion the biodiversity in the environmental education curriculum. So we would like also to know this biodiversity, this biodiversity context is already in the subject or not. That's already in the curriculum or not. So we will like to change like in what subject that you learn about biodiversity. So the option will be like we can have this option again. But that's, and I add all of them because here they have semua, but here there is no semua. So I add that. Okay. So this the question, and the second part is about actually we change it. The question is not about this. It's not about you have to care about biodiversity. Is forgot. You concerned about biodiversity. We change it to be first we care about biodiversity, but again you care about biodiversity is yes or no questions. But this is about opinion. Do you agree or not agree? So we add a term like you have to care about biodiversity. So the question changed to be yes or no. The questions change to be like the opinion. So they can have their opinion. They agree or not. They not agree or not. So that's we try to avoid the yes or no questions. And then your country is a country with high biodiversity. We change it to be your country is rich in biodiversity or mega biodiversity. So just that. This number 4, you effort, because this number 4 is actually connecting to this question, and why we put it here, because to make it less confused, because they are the selecting disagree, agree, and then they have to select again, and then disagree again. So maybe we have to group them, like all the disagree and agree questions put at the first part, and then for the selecting, for the check box, we put it in other part. I mean, they're always two different, but they are really distinguish each other. And that's for me. This we would like to change, like your country efforts in biodiversity protection. So the question to be short is really short. So the question is, are they no? So it's not about the concern of biodiversity. It's about are the student have the knowledge of biodiversity. So the point may be, because after we read this, every question here is related to knowledge of biodiversity, not the concern of biodiversity. And this one somehow the closing of our discussion, because we don't have much time, because it's like mostly every question we really try to take a look of this. The last one is efforts that have been made to protect biodiversity in your country. So the first one we try to make the change to be like establishment, nature reserve or nature conservation area, or you can change. We put two options here. You can choose in situ or ex situ conservation establishment. So you can choose one of these, and because this establishment, nature reserve or nature conservation, in situ and ex situ conservation, it means zoo, flower garden, protecting endangered animal and plants, and then so these three, it should be, you can omit this because it's already taken by the first options. And this is the question: what is industrialization of animals? Because we don't really. First, I cannot explain what is it. Maybe we need to make it clear what is it, what it means. What is the industrialization of animals? And then, we still keep environmental restorations. We still keep prevention of invasive species. And this one, application of biodiversity curriculum in educational environments, which tends to be the integration of environmental education into curriculum. That's maybe from our group. I'm sorry, because there's still a lot of questions. We cannot go there. So that's actually from me. And that's all. Thank you very much. And I'm sorry that we don't really finish it. And one more, sorry, and one more. Maybe we would suggest that this kind of document, this kind of word, maybe you should put it into the Google Doc, and you can share it in the participants, so they can have their input in that document. That's thank you for me. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, sir. And big applause for our moderator. Okay, before closing the regional workshop, we present a certificate appreciation to all moderator. We welcome to Miss Risa Rosita, given by certificate for first moderator, Dr. Suprianto. Okay, Mas Asep, take a picture. Jepret. Oke, bentar Mbak. Handphone gue. Okay. Next, to Dr. Romi Ardiansyah, please. For the second moderator. Okay, big applause for Dr. Romi and Dr. Suprianto. And next, certificate virtual, please, committee. For Dr. Sri Suhartiningsi. Certificate given by Okay, thank you. Okay, next session, we welcome to Ms. Risa Rosita for closing statement and closing the regional workshop in today. Please, Ms. Risa Rosita, time is yours.
Okay, thank you to the MC, respected Deputy Director for Program of Seameo Biotrop, Dr. Evelyn V. Bikas, affiliate scientists, head of sections, and staff of Seameo Biotrop, moderators and participants in Indonesia and outside country. It is indeed a great pleasure and honor for me to be here on the occasion of the official closing of the regional workshop on the inclusion of biodiversity in the environmental education curriculum. Finally, we already finished the workshop from 1 August up to 3 August 2022. This workshop was attended by more than 102 participants from Indonesia, Cambodia, Malaysia, and the Philippines. Of the participants, 8% came from governance, 9% from research agencies, 43% from school, 35% from university, and 5% from private companies. Today, we have already obtained and formulated the questionnaire related to our study of the inclusion of biodiversity in the environmental education curriculum in Indonesia and Asian countries. For schools, referring to the pre-test and post-test, we conclude that this workshop has been successfully implemented because 60% of participants got post-test score more than 75. On the other hand, overall participant satisfaction rate of our regional workshop on the inclusion of biodiversity in the environmental education curriculum is 98.5%. Please let's give applause to all of us here for research persons, moderators, and also our participants. So I am, as the workshop coordinator, would like to express my deep gratitude to our research persons, the committee, and others who collaborate and participated in the regional workshop. Thanks for your input and ideas for us. This workshop would not have been success without them. Hopefully, this workshop can be useful for all of us, and there will be opportunities to collaborate with you in the future. Have a good time ahead to you all, and see you later. Thank you.
Thank you, Miss Rosita. And ladies and gentlemen, the closing statement